I want to tell you about a cool study I found that was recently published in the journal Scientific Reports. The research is focused on a particular kind of stem cell called perivascular stem cells, which can differentiate into either fat cells or bone cells. Essentially, these perivascular stem cells are the precursors of both bone and fat cells. I know that it seems rather odd that the osteocytes, the bone cells that secrete mineral salts to build hard bone, come from the same progenitor stem cells that can also give rise to the soft, lipid-filled fat cells. The progenitor stem cells have an internal mechanism for making and promoting this differentiation, and scientists believe that they've figured it out. The research team is led by senior scientist Aaron James, an associate professor of pathology at the Johns Hopkins School of Medicine. In their previous experiments, Mr. James had identified a protein called WISP-1, or RISP-1, which stands for the WNTI Inducible Signaling Pathway Protein 1. They identified it as a biomolecule of interest in the differentiation of perivascular cells into bone cells. However, the full extent of its involvement in bone cell development was not fully understood. The purpose of this new study was to peer deeper into this phenomenon and figure it out. First, the researchers isolated human perivascular stem cells, and they noticed that they sustained a high expression of WISP-1. Naturally, they wondered what would happen if the WISP-1 gene was knocked out. So they knocked out the gene, and they observed the cells that now couldn't express WISP-1. They observed that the stem cells began to differentiate into fat cells. 4 genes that are involved in fat formation became 50 to 200% more active. For example, the FABP4 gene, which expresses fatty acid binding protein 4, saw a 139.8% increase in activity, and the gene that expresses lipoprotein lipase, which breaks down triglycerides into free fatty acids and monoacyl glycerol, saw a 172.8% increase in activity. In addition, other genes that are involved in bone formation, like the differentiation into osteocytes, those genes became about 25 to 60% less active. For example, the collagen type 1 alpha-1 chain protein, which is the primary protein in the bone matrix itself, saw a 27% reduction in expression. Furthermore, the deposition of bone nodules, or the initial growth points of new bone tissue, saw a, quote, robust inhibition, unquote, when the WISP-1 gene was knocked out. Now, to see how this works in the other direction, they implanted a WISP-1 expression plasmid into fresh perivascular stem cells. This caused these stem cells to express WISP-1, so the researchers could study the emergent phenomena. What they saw was that the stem cells began to turn into bone tissue. They began to differentiate into osteocytes. Various osteogenic gene markers saw a swift uptick in activity, like the RUNX2 gene, which increased 119.1%. Adipogenic gene markers, or genetic activity signaling the growth of adipose tissue, saw a reduction in activity, and after 15 days of this differentiation, there was a significant reduction in the intracellular accumulation of lipids. So they were kind of clearing out the lipids. The cell was saying, I'm not becoming a fat cell, I'm becoming a bone cell. So let's get rid of all of these stores of lipids, of fatty acids. Okay, so this WISP-1 gene really seems to be an integral part of the regulatory mechanism controlling perivascular stem cell differentiation into either bone or fat cells. Now this is where it gets really exciting, because the researchers created a solution with these WISP-1 activated human stem cells, and injected them between the vertebral bones of mice. After 4 weeks, the researchers saw that these stem cells were still producing high levels of the WISP-1 protein, and as a result, there was extensive new bone growth. The bone growth had successfully fused the separate vertebra together. In humans, fusing some vertebral bones together can be a treatment for health issues like back pain, or a slipped disc, or a vertebral fracture, but this requires invasive surgery and the implantation of metal rods. If this new method can be used effectively in humans, it could be a way to perform spinal fusions without the invasive surgery. A traditional spinal fusion, quote, requires a massive amount of new bone cells, said the lead researcher Aaron James. He went on to say, quote, if we could direct bone cell creation at the site of the fusion, we could help patients recover more quickly and reduce the risk of complications, unquote. When their initial experiment showed success, Mr. James said, quote, we hope our findings will advance the development of cellular therapies to promote bone formation after surgeries like this one, and for other skeletal injuries and diseases, such as broken bones and osteoporosis, unquote. So, to summarize all of this, they might have developed a kind of cellular goop that can be injected into an injured bone, where it will ossify and grow into new, healthy bone tissue. This could potentially be a futuristic, sci-fi method of treating bone injuries without any major surgery at all. Simply get a targeted injection of properly differentiated stem cells and do some follow-up screenings to make sure everything's healing nicely, and there you go. Stem cells in general hold incredible medical potential for all kinds of ailments, so it's going to be really exciting to see what new developments come out of this field in the near future.